So, before we begin solving ATP questions, the first thing you need to know is the practical equipment used in biology. Although you will not perform the experiments yourself, you must be able to recognize each piece of equipment and understand its function. The common laboratory equipment you need to remember includes the microscope, hand lens, scalpel, razor blade, forceps, mounted needle, mortar and pestle, test tube rack, measuring cylinder, beaker, pipette, dropper, syringe, glass rod, thermometer, stopwatch, balance, colorimeter, water bath, Petri dish, white tile, microscope slide, and cover slip. Now let's understand the purpose of each one. The microscope is used to magnify tiny cells and organisms that cannot be seen with the naked eye. A hand lens also magnifies objects, but at a much lower power, making it useful for observing larger specimens such as leaves and insects. A microscope slide holds a thin specimen for viewing under the microscope, while the cover slip keeps the specimen flat and protects the microscope lens. For cutting and dissection, a scalpel is used to make clean, precise cuts through tissues. A razor blade is used to prepare very thin sections of specimens for microscopic examination. Forceps are used to hold or pick up delicate specimens without touching them, and a mounted needle is used to separate, point at, or move tiny structures during observation. Several pieces of equipment are used for handling liquids. A test tube holds, mixes, or heats small amounts of liquids, while a test tube rack keeps test tubes upright and organized. A beaker is used to hold, mix, stir, and heat larger volumes of liquid. A pipette transfers accurate measured volumes of liquid, whereas a dropper is used to add small drops of liquid without measuring them. A syringe is used to draw up or transfer precise amounts of liquids or gases. Some equipment is used for measurement and temperature control. A measuring cylinder accurately measures the volume of liquids. A thermometer measures temperature during experiments. A stopwatch measures time accurately. A balance measures the mass of substances, while a colorimeter measures how much light passes through a solution to determine its concentration. A water bath gently heats samples while maintaining a constant temperature without direct heating. Other equipment is used for preparing specimens. A mortar and pestle crushes solid materials such as plant tissues into a fine paste. A glass rod is used for stirring liquids and mixtures. A Petri dish is used to culture microorganisms or hold small specimens, and a white tile provides a clean background for observing color changes or cutting specimens. Questions that simply ask you to identify laboratory equipment are quite rare. However, if the examiner gives you an experiment or practical investigation, you should immediately be able to recognize which of these instruments are required and why they are used. Knowing the purpose of each piece of equipment will make practical questions much easier to answer. Now, take a close look at each of these instruments. They all have different shapes and appearances, so make sure you can recognize them easily if they appear in the examination.